Hi everyone, my name is Brooke Dunnery and I am currently in my third year at Coastal Carolina University. Last year, I took organic chemistry with Dr. Brian Wakefield and fell in love with the subject. He conducts research on campus and was willing to let me join his laboratory. Under his guidance and the guidance of a fellow Wakefield group member, James Heldman, I have completed a series of reactions to synthesize a Phidianidine analog that contains a 2,4-disubstituted oxazole ring in place of the 1,2,4-oxadiazole ring that is found in naturally produced Phidianidines. So what are Phidianidines, and why is our research laboratory interested in studying them? Phidianidines are naturally produced compounds that were originally isolated in 2011 from a species of marine sea slug, Phidiana militaris. An image of this marine sea slug is pictured in the lower right-hand corner of this slide. The structures of the two originally isolated Phidianidines, A and B, are shown in the lower left-hand corner. Highlighted in yellow is a part of their structure called a 1,2,4-oxadiazole ring. Our research laboratory is interested in studying this natural product because many natural products do not possess a 1,2,4-oxadiazole ring. Furthermore, Phidianidine analogs exhibit important biological activities, such as cytotoxicity of cancer cells, selective inhibition of central nervous system targets, neuroprotective properties, anti-cancer properties, and immunosuppressive properties. Because previous studies have indicated that the two naturally produced Phidianidines, each containing a 1,2,4-oxadiazole ring, exhibit biological activity, one research focus of our laboratory is to study and determine the biological activity of Phidianidine analogs that contain an oxazole ring. Structurally, the only difference between an oxazole ring and an oxadiazole ring is the number of nitrogen atoms present. An oxazole ring contains one nitrogen atom, whereas an oxadiazole ring contains two nitrogen atoms. For your reference, images of these two rings are shown at the bottom of this slide. In order to study the biological activity of a Phidianidine analog that contains an oxazole ring, James and I are working on determining a specific pathway for the total synthesis of a Phidianidine analog that contains an oxazole ring in place of the 1,2,4-oxadiazole ring that is found in naturally produced Phidianidines. We are conducting this research to determine whether or not the oxazole ring, or the absence of the second nitrogen atom, has an effect on the biological activity of the analog, and to determine the analog's potential to be used as a therapeutic agent against cancer, neurodegenerative diseases, and other diseases. As previously mentioned, I have been able to complete 6 reactions in order to synthesize a Phidianidine analog with a 2,4-disubstituted oxazole ring, under the guidance of Dr. Brian Wakefield and fellow Wakefield group member James Heldman. The first reaction that I completed in this synthesis was a Suzuki-Miura coupling in water, the first reaction pictured on this slide. In this reaction, 5-bromofuran 2-carboxylic acid was reacted with 4-ethoxyphenylboronic acid. The percent yield of this reaction was 52, which may not seem high, but it was actually the highest yield I received out of all of the 6 reactions in this synthesis. Next, a peptide coupling reaction was performed in which the product of reaction 1 was reacted with a serine methyl ester. This produced a product with an amide functional group. The amide product from reaction 2 then underwent a cyclodehydration to form the 2,4-disubstituted oxazole ring. Reaction 3 took place in 3 steps, as noted by the 3 steps near the reaction arrow, and had a percent yield of 37. Since the structure of the product of the cyclodehydration reaction, reaction 3 on the previous slide, was verified via NMR spectroscopy results, I was able to continue this synthesis. In the 4th reaction, the first reaction you see on this slide, the ester on the 4th position of the oxazole ring was converted to a carboxylic acid. This reaction had a percent yield of 37. In reaction 5, I used oxalyl chloride to convert the carboxylic acid functional group to an acid chloride. I then introduced indole via Friedel-Crafts acylation in reaction 6. This reaction had a percent yield of 34%. Since the desired Phidianidine analog does not contain a ketone, I must determine reaction conditions for an additional 7th reaction that will reduce the ketone that forms after the Friedel-Crafts acylation. I would like to close my research presentation today with a few conclusions and a few thank yous. My first conclusion is that my reactions were somewhat successful. What I mean by this is I successfully completed reactions 1, 2, and 3 based on the NMR spectroscopy results for the product of reaction 3. However, my yields were not good. Additionally, I have yet to verify the structures of reactions 4, 5, and 6, and I am hopeful that this spring I will be able to verify the structure of reaction 6 using IR spectroscopy. In the future, I would like to try using different reagents and catalysts to improve the yields of the individual reactions in this synthesis. So the next steps for my research are to verify the structure of the 6th reaction with IR spectroscopy, and if the desired structure was formed, to try similar synthesis to add indole rings with different substitution patterns, to optimize the yields of individual reactions in this synthesis, and to find reaction conditions to reduce the ketone that resulted from the Friedel-Crafts acylation in reaction 6. So I would add a 7th reaction to this synthesis in order to get the final desired structural analog. Thank you for listening to my presentation today. I really hope you enjoyed it. I wanted to say a huge thank you to South Carolina INBRE for running this symposium and for providing students with a safe and virtual environment to present their research. I would also like to say a huge thank you to Dr. Wakefield and the Wakefield group members for allowing me to conduct research and help me learn more about the field of organic chemistry. And lastly, I'd like to thank the Gupta College of Science at Coastal Carolina University for their continued support.